Good afternoon, everyone. My name is Wang. I'm a PhD student here at the University of Guelph. It's really just, very inspiring to hear the talk and just really intimidating because I'm talking about some work that I do that doesn't include any innovative technology or anything. But I really am a population ecologist in training. So from that lens, eDNA to me is a way of getting some sort of index that can be used to infer species abundance. But before we do that, before we can reliably trust the eDNA results to infer species abundance, we need to first answer some fundamental questions. And for me personally, I'm really interested to know how does different kind of energetic processes, such as metabolic activities, varying nutritional levels, and life history events interact to shape intraspecific eDNA deposition rates on an individual level, and how does that change throughout individual lifespan. So to do that, we use the classic Daphnia algae system, where we have all of the Daphnia grow in a 2x2 factorial design with two temperatures, 15 and 25, at high and low food. And in order to get that individual shedding rate, all of our Daphnia is raised in a 13 ml glass vial. Oh, that didn't work. Okay. That didn't work either. Okay. As you can see in the glass vial with unique ID that we keep track throughout individual lifespan. So briefly, what we're doing is every day before data collection starts, we put all the Daphnia in its own vial pre-sterilized with the food. We let them stay there for 24 hours. After which, we remove the Daphnia, and then we take individual measurements for all of the Daphnia, including body size, reproductive status, the number of eggs if they're pregnant, or things like whether they died or molted, which is shedding of their exoskeleton during the 24 hours. And then we take the water on the side, we collect 10 mils out of the vial and mix it with ethyl and sodium acetate and let them precipitate at -20 until extraction. And we used QIAGEN blood and tissue kit, and we quantified all of our eDNA samples using droplet digital PCR with a previously established assay designed in the lab by Daniel Borg, right over there. So what do we see? Well, the first is here I have a figure to show you our metabolic model. So we have our log abundance of all of our eDNA copies on the y-axis. On the x-axis, we have body mass, and then we can see that consistent with our metabolic predictions. Larger individuals produce much more eDNA, as you can see by the positive slope. And in addition, there is an interaction between body mass and temperature, suggesting that larger individuals respond to temperature change more rapidly than smaller individuals. And each of the dots here, as you can see, so the 25 is in red and the 15 is in blue. Each of the dot is one individual measurement for one Daphnia over the 24 hours. And then when we take the residual and plot it against different food levels, we see that individuals at high food level produce substantially more eDNA than they were at food level, than they were at low food level, suggesting that feeding and waste excretion is likely the main source of eDNA, at least in our system. And when we compare everything together, because we really wanted to know how does all of those different energetic processes play out, we have our full model shown in this regression coefficient plot. So the yellow is for metabolic parameters, the blue is nutritional, and the purple is life history. And then we can see that we see the same patterns for our metabolic and nutritional. But among our life history things, we see reproductive status played a big role in driving eDNA shedding rate on an individual level, meaning that when the individual are pregnant, they just produce more eDNA. However, the number of eggs on their back didn't actually make a big difference. So it's actually the reproductive state rather than the magnitudes of reproduction, in this case, that matter. We did see a small elevated reproduction eDNA deposition from mortality, showing that recently deceased individual might produce more DNA through deposition than live alive individuals. But of course, we can think about things like different sampling techniques and even environmental RNA that can be used to help mitigate some of those effects. And then we used AIC and BIC to find out this full model is the best. So what does this tell us? Well, we showed that Daphnia deposition rate depends on simultaneous processes. We found out that metabolism is really important. That alone explained over 30% of variation among all of our data set. and that we identified large and pregnant Daphnia had the highest eDNA deposition rate, especially under warmer conditions with high food levels. And therefore, we cautiously conclude that metabolically active individuals should be prioritized for better field detection. But of course, all eDNA studies should be taken with a grain of salt, and context is really important. And results from benchtop experiments should not be overgeneralized because it lacks the complexity of a natural setting. So the next step really is to see how does our data play out in a small mesocosm experiment. And this is something we did last summer where we run Daphnia algae in a 140-liter tank rather than a 13 ml vial, and with my 2 students happily working alongside, and hopefully we'll have more coming up in a few months. And with that, thank you for your time, and thank you for everyone in this class.